Hello, my name is Pedro Torres, and today I will be talking about the different mechanisms used by pathogens in order to escape from the immune responses. As we already know, the immune system successfully recognizes, consumes, and destroys all types of pathogens that are present in any kind of infection. This is done so the pathogen cannot establish itself in the body and cause a horrible disease that may not be cured or may require a lot of medication in the future. However, the immune system does not always succeed in its job. Often, the reason for its failure is due to the invading pathogens that have evolved strategies for evading or suppressing the host's immune response. The evasion mechanisms. Many different mechanisms have evolved in different organisms, but today I will only be talking about three common mechanisms used, which are escape killing, antigenic variation, which in this case the pathogens alter their surface protein to evade the immune response, and latency, which in this case existing but not yet been well developed. Escape killing. First, I will start by talking about the strategies used by pathogens in order to escape death. Molecular mimicry is commonly used in pathogens in order to genetically resemble parts of the host's own body. When the pathogen mimics the chemical makeup of the body, they can exist without an immune response being activated against them. So they can move around in the host, without any problems, without being noticed by any of the antibodies. Okay. For example, it's like when a criminal kills an innocent person and the police are looking for him, and find someone that looks exactly like him and arrest him for the crime they didn't commit. It's the same way in molecular mimicry. As you can see in the picture below, we got the pathogens. They all look alike, and the immune response will arrest one of them because they think one of them is the killer. Some pathogens can express protein epitopes that resemble self-components in either conformation or primary sequence. For example, in rheumatic fever, a disease that is caused by autoimmune destruction of heart cells can develop after a group A streptococcosis infection. In this case, antibodies to streptococcal antigens have shown to cross-react with heart muscle protein that result in immune complex disposition. Now I will begin by talking about hiding, how the pathogens can escape this way. Many pathogens avoid being detected in an immune response by hiding inside the cells of the immune system. When doing this, they will not present antigens that will start an immune response. They will multiply inside the cells and later invade the body when they generate a large amount of them. Among the bacteria that use this strategy are Brucella, Listeria, Mycobacterium tuberculosis. They all infect the macrophages. As you know, the macrophages are cells that are normally responsible for destroying these invading bacteria. Two other ways pathogens can escape from being killed is by escaping to the cytoplasm or coating themselves with proteins. As we can see in the first example, we got Listeria monocytogenes coming into the cell and going out to the cytoplasm. The reason they do this is because the lysosomes are surrounding the Listeria monocytogenes, and they go out into the cytoplasm so they can replicate. As we can see in this picture below, this is Listeria monocytogenes replicating and getting ready to move on. The second example is Treponema pallidum. They coat themselves with proteins so they won't be recognized by the antibodies and get killed. Now I will begin by talking about how antigenic variation is used by infectious agents to evade the immune response. In antigenic variation, the extracellular pathogens alter their antigens. The mechanism is important for these pathogens so they can target long-lived hosts and repeatedly infect it. Antigenic variation not only enables immune evasion by the pathogen, but also allows microbes to cause reinfection, as their antigens are no longer recognized by the host's immune response. For example, it's like when antibodies are the police and they go out there and look for the pathogen, which is the criminal. But the pathogen changes its shirt in order to not be recognized, and it's harder to find it this way. There are three ways in which antigenic variation can occur, but I will only be talking about two. Uh, for example, 84 known types of Streptococcus pneumoniae are an important cause of bacterial pneumoniae. Each type is different from its structure because of its polysaccharide capsule. As we can see in this picture is a Streptococcus pneumoniae with a polysaccharide capsule. The different strains of Streptococcus pneumoniae have antigenically distinct capsular polysaccharides. As we can see in this picture right here, this diagram, we got the Streptococcus pneumoniae, different types with different, they're the same, but they have different capsular polysaccharides within them. The capsule prevents effective phagocytosis until the bacterium is opsonized by specific antibodies. When someone is infected with Streptococcus pneumoniae, the response is clear. The response clears the infection, but then another strain of Streptococcus pneumoniae is produced, and another response has to be created, and it goes on and on. Another mechanism of antigenic variation is seen in the influenza virus. Many years ago, this virus was responsible for most infections throughout the world. As we can see in this diagram over here, these are all the influenza virus types that have happened in the past years. 1918 was one of the deadliest, also called the Spanish flu. The human population gradually develops protective immunity to this virus type uh, by directly neutralize antibodies against the major surface proteins of the influenza virus, which is a hemagglutinin. Hemagglutinin is responsible for viral binding to and entry into the cells. Two types of variation allow repeated infection with type A influenza virus. The first of these is antigenic drift, which is caused by a point mutation in the genes encoding hemagglutinin in the second surface protein, neuraminidase. The progressive mutation changes the structure of the hemagglutinin and neuraminidase spikes, It is slow and a steady change. So we can see hemagglutinin uh, mutation structure has been changed in the neuromine days of the spiking. Also down here, neutralizing antibodies against hemagglutinin block the binding cells. The mutation alters the hemagglutinin, so the neutralizing antibody no longer binds. Major influenza pandemics have resulted in widespread and fatal disease have occurred as a result of the second process, which is termed antigenic shift. This happens when there is a recombination of the segmented RNA genome between different influenza types that will lead to major changes in the hemagglutinin protein on the viral surface. The resulting virus is poorly recognized by the antibodies and T cells directed against the previous variant, so that most people are highly susceptible to the new virus and severe infection results. So we can see an antigenic shift. Um, the RNA segments are interchanged between viral strains and secondary hosts. No cross protective immunity to the virus expressing a novel hemagglutinin. A third mechanism used by pathogens in order to evade the immune system is latency. Latency is the ability of a pathogenic virus to lie asleep within a cell, denoted as the lysogenic part of the viral life cycle. During the latent state, the virus does not cause disease, but because there is no viral proteins to stop its presence, the virus cannot be eliminated. Many latent infections can be reactivated, and this results in recurring illnesses. For example, almost all herpes viruses often enter latency. Herpes simplex virus, the cause of cold sores, infects epithelia and spreads to the sensory neurons, serving to the area of infection. After an effective immune response controls the epithelial infection, the virus persists in a latent state in the sensory neurons. Factors such as sunlight or bacterial infections reactivate the virus, which then travel down the axons of the sensory neuron and reinfect the epithelial tissues. During this stage, the immune response again become active and control the local infection by killing the epithelial cells and produce a new sore. The cell cycle can be repeated many times. There are two reasons why the sensory neuron remains infected. First, the virus is quiescent in the nerve and therefore new viral proteins are produced. Neurons carry very low levels of MH class 1 molecules, which makes it harder for CD8 T cells to recognize infected neurons and attack them. As we can see in this diagram, herpes virus enters the body, it then goes and lies in the nerves, then the herpes virus is reactivated, causing an outbreak. It will then cause blisters in the skin to reoccur. These were only three common ways pathogen invasion occurs and lets us know how the immune system can fail because of its weaknesses. And these are three of the references that I used for this video. Thank you for watching.